Life writes the questions. We look for the answers. Since its founding in 1896, Roche has grown from a tiny drug maker into one of the world's leading healthcare companies. From the start, founder Fritz Hoffmann-La Roche's focus was on innovation and building a company with an international presence. In the last 100-plus years, Roche has pioneered many medical breakthroughs. Our investments in research not only earned us Nobel Prizes, but also laid the foundation for some groundbreaking therapies in oncology. Today, Roche contributes on a broad range of fronts to improving people's health and quality of life. We focus on disease areas of high, unmet medical need. Roche's Basel headquarters continues to be one of the group's biggest sites for research and development, manufacturing and administration. To this day, it remains a key operational hub within the Roche Group. Every treatment starts with a diagnosis, and the more accurate the diagnosis, the better the chances of providing effective therapy. With its combined expertise in both diagnostics and pharmaceuticals, Roche is uniquely positioned to tailor treatments to specific patient populations. Drawing on advances in molecular biology and pharmacogenetics, Roche is moving closer and closer to making personalized healthcare a reality. The drug development process can be broadly divided into 6 steps. 1. Identifying a molecular drug target. 2. Identifying a first lead compound. 3. Lead optimization. 4. Preclinical safety and efficacy trials. 5. Clinical trials in man. And 6. Regulatory approval and launch. Step 1. Identifying a molecular drug target. To fight a disease effectively, it's important to understand how people who have the disease differ from those who don't. Researchers need to understand the underlying mechanisms of the disease as fully as possible. Molecules and the pathways that influence the development or progression of a disease are potential drug targets. Step 2. Identifying a lead compound. The next step is to find a drug that will act on the target in the desired way. Basically, there are 2 options here. Researchers can look for a small chemical compound that will do the job. Most of the classic medicines of the past fall into this category. Or they can look for a large biological molecule. Therapeutic antibodies are a common type of biological molecule. Many of today's cancer medicines belong to this class. In small molecule chemistry, the challenge is to discover the right compound amid a limitless range of possibilities. The Roche Compound Library, located in Basel but accessible to Roche scientists worldwide, contains more than 1 million different compounds. And each year, medicinal chemists throughout the Roche Group synthesize thousands more. Automated high-throughput tests are often used to identify first lead compounds with the desired activity. These tests screen thousands of compounds in a very short time to identify those that interact with the target of interest.